So we do everything what they are doing.
Hello, hello,
That's
check,
the problem.
check mic. Hello,
वो टेक्निकल कल्चर में शिफ्ट
hello,
हम लोग की वो कल्चर में शिफ्ट होने को क्योंकि
hello, check mic. Hello, hello,
मानते हैं जो कर रहे हैं
hello, check mic. Hello, hello, hello,
थोड़ा सा डाइजेशन
Hello.
प्रॉब्लम
check. Hello, 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 check. Hello, 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 hello,
अच्छा बहुत बढ़िया एक्सेलेंट
hello, 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 check mic. Hello,
When
okay.
we hear such people, it seems like they have a young brain. यहाँ क्या है कि हॉकिंग साइंस है अब AI से साइंस हो गया है नेचुरल इंटेलिजेंस खत्म हो गया
अभी तो नहीं कर पड़ेंगे पहले ना कैलकुलेशंस करते थे हाथ से करते थे
जी 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 अब कैलकुलेटर से अब कंप्यूटर से करते हैं
कैलकुलेटर अभी इवन 2x2 करना है तो भी कैलकुलेटर आते हैं
नहीं कंप्यूटर से करते हैं
तो इस तरह से अभी क्या हो गया कि AI के चक्कर में
नहीं नेचुरल खत्म हो गया ना नेचुरल खत्म हो गया यहाँ आर्टिफिशियल इंटेलिजेंस आया नेचुरल खत्म हो गया
आपको अपना दिमाग भी यूज़ करने की ज़रूरत नहीं है
पहले इसको यार बंद करेंगे
समिट की तो पूछा कि आपने AI सपोर्ट लिया है कि नहीं
पूछता जोशी जी से DBT DST में मैंने ओपन फोरम सिग्नेचर है अगर प्लांट है तो अंडरस्टूड है कि सब फोटोसिंथेसिस करता है हम क्राफ्ट प्रोडक्टिविटी की बात करते हैं फूड की बात करते हैं हम फोटोसिंथेसिस करता है
We will begin our session shortly.
हमारा काफ़ी बच्चा है
आपका रिप्रेजेंटेशन बहुत ज़्यादा है
And then over to
कितने बजे शुरू करना है 10:15
Five more minutes.
धारा कहाँ है उसको बुलाओ जरा नहीं वो तो क्या है
अभी तो कहीं जाना अभी तो नहीं नहीं टाइम नहीं है काम क्या होता है
नहीं
तो एग्जाम
पता है
सही बात सर ये नहीं है सही बात ये कि हो नहीं आपके 30 स्टूडेंट
यहाँ से
यही तो अभी यहाँ पे नहीं हुआ
आप आए
वो ब्रेकफास्ट आ रहे बस
अभी इसी बार भी 30 ऑलरेडी फैक्टर थे तब भी नहीं होता है
क्या थी
वो लोग की यही तो सरप्राइजिंग है
वो बाद में बताएंगे ये तो जो भी है उसका वो था क्या कहते हैं एक कमेटी होती है BRC उसका मेंबर 3 साल तक उसको ले लिया ये जो टाइफाइड वाले जो मीटिंग किए थे उसका कुछ आगे रहा कि ये लेकर बैठ गए।
क्या बोलते हैं फाउंडेशन कुछ सिर्फ नाम के लिए उसमें से बोलता है
एक्चुअली
बात ये सब बंद कर देंगे DBT GST
तो ये देखा ग्लोबली इनके केवल पेपर नहीं रहते, इनको हर चीज़ मैंने इम्पोर्ट बाहर से करना है, हाथ से इम्पोर्टेंट है। अगर फ्रांस में 90% क्या दिया ये तो हम दिखा, लेकिन वो नहीं देखो कि चाइना जो है कितना फ्लश कर रहा है, नहीं कितना फ्लश कर रहा है। वही मजबूरी
साइड वाला
साल के लिए एक्सटेंशन देते हैं। वहाँ क्या हुआ डायरेक्टली एसोसिएट प्रोफेसर के इक्विवेलेंट है GBRC, इसलिए कोई सीनियर आदमी आते हैं
पेन से लिए तुम
इसलिए तो मैं उसमें जाती हूँ आपको बताऊँगा डिटेल्स गलती हो गई।
जैसे 4 लोगों को रखा है, 4 लोग जो है 20 सिंपल है, फिर जिंदगी नहीं समझती।
अभी उनकी पॉलिसी नहीं देनी पर एक बार प्रोजेक्ट देनी
नहीं अभी नहीं वही तो फाइनेंस का बजट किया है, मेरा कम बोल रहा था
क्या
इसका प्रेजेंटेशन वो करके आए हैं शुरू कर लो।
आप तो वो पॉलिसी इसलिए अभी
2-3 महीने
गिविंग द कॉन्फ्रेंस के लिए भी बैठा नहीं
2-3 महीने ये तो मिला
पैसा है गुजरात में तो जेल भी
A quick and humble reminder, please keep your mobile phones on silent mode. A very good morning to all. I can see fresh faces after a very good night's sleep. Nevertheless, on this beautiful morning, I'm pleased to welcome you all again to the another exciting and wonderful meet of this 2-day national conference on antimicrobial resistant trends, environmental microbiology and industrial solutions. As it is rightly said, days are not lost when memories are created, and let us all move ahead and add more to it. I hope you have enjoyed the day one of A.R.T.E.M.I.S to the fullest. I hope it. Isn't it? Yes or no? Let's have a big round of applause for yesterday's event. Great. So with an immense pleasure, I welcome Professor Datta Madamvar, Scientific Advisor, Charotar University of Science and Technology, Anand, for our third invited talk of A.R.T.E.M.I.S 2024. Professor Madamvar obtained his PhD from BITS Pilani. He is a former professor and head Department of Biosciences and former Dean Faculty of Science at Sardar Patel University, Vallabh He worked as a postdoctoral fellow at TIFR, Mumbai, University of Frankfurt, University of Constance, and also served at BITS Pilani. Professor Madamvar is a microbial biotechnologist with diverse research interest. His main research focus is on microbial bioremediation, environmental biotechnology, non-aqueous enzymology, and cyanobacterial biotechnology. He is a recipient of European Commission Visiting Scientist Fellowship, Visiting Fellowship at Swiss Federal Institute of Technology, Switzerland, Visiting Professorship at University of Blaise Pascal, Clermont-Ferrand, France, Lifetime Achievement Award of BRSI for the year 2019, Lifetime Microbiology Devotion Award by Microbiologist Society India for the year 2021, Lifetime Achievement Award for Association of Biotechnology and Pharmacy for the year 2022, Fellow of International Bioprocessing Association, Fellow of Biotech Research Associate of India, Fellow of Association of Microbiologists of India, Fellow of Association of Biotechnology and Pharmacy and Gujarat Science Academy, BHU Senatorial Award of BRSI, and member of several academic bodies, member of task force, expert committees of various agencies such as DBT, DST, GSB, TM, etc. He is also a member of editorial board of several national and international journals. Professor Madamvar has published more than 290 research articles, several book chapters, and one provisional American patent issue of credit. He is having an H index of 70 and I-10 index of 215 with more than 16,500 citations. Professor Madamvar is listed in world ranking of top 2% scientists. He is a well-traveled researcher with his research visits and gave invited talks in several countries including Germany, UK, Austria, Switzerland, France, USA, Malaysia, Singapore, Brazil, China, Dubai, Finland, Japan, and many others. Please give a big round of applause. So let us all invite Professor Madamvar, an admirable personality, on the dais for his technical session. And before that, I would request Professor Buddhisagar sir to please greet our guest with a bouquet. तालियां बजती रहें.
Very much, sir. Very nice, sir. Thank you. Thank you.
Color like that.
एक स्लाइड मूव करने वाला
Slide.
Thank you very much for such a nice word. I didn't know that I do have that. Thanks a lot. First of all, let me thanks the organizing committee, both Professor Tiwaris and their colleagues. Well, this was the talk which I did earlier also gave to the IIAR, but that was through online. As it was rightly pointed out that there are three major areas where I get involved. One of them happens to be cyanobacterial biotechnology. Other are the non-aqueous enzymology, where you can make use of enzyme in organic solvent, yet they don't lose their activity. And thirdly, that is the environmental biotechnology, where we try to go for the treatment of the industrial liquid waste. And we try to understand the microbial diversity, how these microorganisms are playing a role in the treatment process. Well, cyanobacteria are considered to be the oldest branching group of prokaryotes. And it is believed that, rather I would say that, they carry out the oxygenic photosynthesis like higher plant, and it is believed that the present oxygenic condition is mainly contributed by the cyanobacteria. They are found everywhere, not necessarily only in the aqueous environment, even in the terrestrial region also. They are also present under the extreme conditions such as polar regions, hot spring, saline desert, and so on. And since they have been involved almost about 2.5 billion years before, over a period of time, they have faced various abiotic situations. And this has positively pressurized the cyanobacteria, and they are evolved in such a way that they produce such compounds which are able to cope up such stress conditions, and that includes the production of pigments, production of certain other molecules like microsporin like amino acids, which can provide you the UV protection, something like that. So like that, there are various compounds which are produced by them. They carry out the photosynthesis. Actually, let's say, what we try to do that in our lab, we maintain large number of cyanobacteria which have been procured from the different environmental conditions. And these cyanobacteria are exploited basically for two major reasons. One is, we try to isolate the phycobiliproteins from these, and they are purified to highest extent. Once they are purified, we go for the crystallization, and then followed by the X-ray diffraction, and thereby you can get three-dimensional structure. And then try to correlate with the function. And these have lot of therapeutical properties which we will see later on. So we try to understand about the therapeutic aspects of these proteins. Similarly, we try to exploit the other molecules which are very small molecules which are called microsporin like amino acids, which have UV protections, which can be used in the for wound healing. It can also provide the skin lotion, something like that. So they are also being tried by us. These cyanobacteria, they carry out the photosynthesis similar to the higher plant in terms of oxygen evaluation. The overall mechanism also having the same, whether it's having the photosystem 1, photosystem 2. The major difference which is found between the higher plants and the cyanobacteria, in case of higher plants, they are able to absorb photon energy in a narrow range, ranging from almost about, you can say, 670 to 720, and that is mainly because of the chlorophyll. Whereas this absorbing window is expanded significantly by the cyanobacteria, and that is mainly because they contain some specific pigments. We call them phycobiliproteins, and these phycobiliproteins, in association with the chlorophyll, they are able to absorb the light radiation starting from almost about 450 to 720, and therefore they have better. So you will find that there are cyanobacteria which will be growing on the surface of the water. There are cyanobacteria which are found in the submerged condition, and you will also find some manufacturer at the bottom of the water. So like that, depending upon the intensity of the light, accordingly they can manipulate, and that is mainly because of the pigment what we have, that's called. What are these phycobiliproteins? The phycobiliproteins are the family of brilliantly colored fluorescent proteins. They mainly consist of two polypeptides, we call them alpha and beta, having the molecular size somewhere around 20 kilodalton, having about 160 to 180 amino acids. These two alpha and beta polypeptide chains, they have a different sequence of amino acid, yet they show the similar bonding pattern, and these together joined together form a dipeptide, which are responsible, and in addition to that they contain what is known as chromophore group, and this chromophore group is attached to the cistinyl residues, which are responsible to provide the color. So you can see that, and they have, these are covalent. And this is in general, in this phycobiliprotein, basically we talk about three different types of phycobiliproteins. We call them phycoerythrin, phycocyanin and allophycocyanin. And depending upon them, accordingly, for example, if I say that PE, that is phycoerythrin, which is a pink in color, phycocyanin and APC, both of them are blue in color, one of them is aqua blue, that is APC, allophycocyanin, and they have this typical fluorescence as well as the absorption spectra, you can see. Like any other protein, obviously you have a corresponding gene, so that particular protein is produced, and you have the heme compound, which is a precursor from where the various chromophore groups are synthesized, and with the lyase enzyme, again they join together and they form the functional molecules. Though these pigments is a dimeric compound, but in a real situation they form a very giant complex, and this giant complex we call them phycobilisomes, which is found on the thylakoid membrane. Now what actually happens when you talk about allophycocyanin, phycocyanin and phycoerythrin, where each one of them is made up of, there is alpha chain, there is a beta chain. Alpha beta chain, which are already appended with the chromophore group, joined together form a heterodimer. And these heterodimers, three such heterodimers again combined together form a trimeric molecules. And in case of phycoerythrin and phycocyanin, these trichomeric molecules further get attached back to back and form a disc. So we call it cylinder disc, something like that. And in case of allophycocyanin, they may not form a but then these trimers get stacked around each other. With the results, what actually happens that at the bottom you have a allophycocyanin which acts as a core cylinder, and from this core cylinder radially, or you can say perpendicularly attached other molecules or the phycoerythrin and thereby it forms a complex. In addition to this chromophore and this word, there is something called linker protein, which is a non-color protein which is only responsible for maintaining the structure. So it has a structural role. This is what happened. Now this phycobiliprotein have several applications. It can be used in the food industries because of their high fluorescent color, non-toxicity, and because of that, you can see that different types of colors can be obtained through this. It can be prepared in the ice cream, it can be in the various soft drinks and various desserts, it can be used, and that can be. In addition to that, it also has a pharmaceutical roles. You will find that many of these phycobiliproteins, they have anti-inflammatory, neuroprotective, hepatoprotective as well as cholesterol reducing ability. So you have several. In addition to that, these days they are also being used in the lipsticks and other things, various cosmetic compounds. Plus, you will see later on that they also have a role in various neurodegenerative diseases. So it can have therapeutic application. At the same time, they being highly antioxidant in nature, they can be used as a nutraceuticals, and plus like green fluorescence marker the way we use, similarly you can also make use of this pigment as a green fluorescence marker, not green fluorescence marker. So there are n number of applications one can make use of it. So what we are trying to do in our case, we try to purify the protein from the cyanobacteria. Once the protein is purified, we try to get the gene corresponding to that, do the nucleotide sequencing of the genes, so you have the whole sequence available, and with the help of that, and then we also do the section. And then we have to fractal what you call the sequencing by using the MALDI, and then document. So we can able to get the 3-dimensional structure. Once we have 3-dimensional structure, you can also do the what is called the docking study, and then there are various other applications which we have to do. And to do that, you require to develop a protocols for purification. These are all conventional protocols. We go for the whether it is a salt precipitations or the what you call solvent precipitations, followed by the ion exchange chromatography, molecular shift chromatography, and the various other things. So in a sequential manner you go ahead with that. What is important that these are all conventional known methods, but what you require to do, you require to do little bit of modification. You require to do certain changes. You require to add some new molecules in that. Perhaps that will improve the efficiency of the purification. There is no standard protocol which is found to be universal for all cyanobacterial protein. If I have a cyanobacteria which is, let us say, Anavina, my protocol will follow this. If I have another organism like Formidium, the protocol, the same protocol may not be applicable for this. I will have to go for little modification of that. So that's how you have to. So you keep doing those various changes, and that's how we have gone. Some of the important thing is that one of the cases we have used the tritonic surfactant. By adding this so-called non-ionic surfactant, your resolution has helped. The separation of phycoerythrin from phycocyanin and allophycocyanin becomes easier. So you have helped them. Similarly, in case of allophycocyanin, we have used specifically ethodiene molecule, and that has helped to separate the allophycocyanin. So like that you can keep on doing that, and you will be able to do, and that's how we go ahead, and that's several protocols have been published. One of the molecules which was initially purified, that's called phycoerythrin from the Formidium species, and when we purify that and we went for the crystallization after getting the X-ray diffraction studies and as well as partial amino acid sequencing, we noticed that this molecule is devoid of 31 amino acid from the N terminal. So it is a truncated molecule. At the first glance we thought that it is a non-functional molecule. And since it is a non-functional, it will not go for dimerization, it will not go for the formation of the complex. But later on we noticed that this molecule is still functional. So those 31 amino acids which were removed from here, they were basically not involved in the functional category, rather they serve as a nutrient, they serve as a nitrogen source for the cyanobacteria itself. That means that if you allow the cyanobacteria to grow in a specific medium, and if you extend the incubation for a longer period of time, the stage will reach where the nitrogen content is completely lost. So in that case, they will use. So this also serves as a sort of a nutrient for them, and that's what it happens. And we could get that. And this was the first paper on the truncated phycoerythrin, which was crystallized, and it was reported, and it was not only from our lab, but it was the first time from the India this was reported. Similarly, we have another organism called leptolingmia, and from this leptolingmia we purified phycocyanin, and this phycocyanin, following the similar protocol, we could go for crystallization. We got the X-ray diffraction data, and after getting the X-ray diffraction data, we could derive the structure. And it was noticed that, so then obviously we went for the docking study, how it would happen. So we thought that there is an enzyme known as beta secretase. All of you may be aware about that. And beta secretase is an enzyme which involved in the proteolytic cleavage of hematopoietin. So it results into the plaque formation. It is a stage which involved during the Alzheimer. So beta secretase is one of the target molecule which can be blocked. So as such, for Alzheimer, we don't have a very clear-cut medicine available. You can slightly delay, you can slightly. So there are some molecules which are already in the market which work as a target to the beta secretase. Through the docking study, we try to see that how does our molecule is it, and we noticed that to our surprise that it has a better affinity to the beta secretase than already existing molecule. So it means that this protein has a better ability to block the beta secretase activity, and that's what. So it has a potential use in that. We'll see that later on how we did further. In fact, what we did it not only for the phycocyanin, we got another molecule called phycoerythrin, which was also crystallized. We also derived the structure of that. At the same time, we got the beta secretase protein from the human gene. So we got the human gene, beta secretase, that was expressed in the E. coli, and since it was placed in the E. coli, it did not go for the expression. So many of you may be knowing that there are a lot of difficulties when you have a expression of the higher organism eukaryotic system into the prokaryotic system. So we went for the codon optimization, and there is a company in the Germany called Gene Art who did the codon optimization for this. We optimized the codon, and after optimizing the codon, that gene was placed under again the E. coli, and it was expressed. And unfortunately, there also, even after the expression, it went into the inclusion body. So what we did, we tried to go for the, again using the what is called denatured and naturation conditions, we tried to get the protein from that. And protein we got it, and then again we went for the crystallization of that protein, we got the structure of that protein, and meanwhile we did the protein-protein interaction with SPR studies. And we noticed that it has got a very strong affinity toward each other. And we were to go for the co-crystallization, having the both protein together, and wanted to go for co-crystallization, but unfortunately that was the fag end of my life in the Sardar Patel University, and there was a big fire in the lab, and we lost everything. So anyway, whatever it may be, but that's all. But then you can think about using that type of activity. Okay. Then we have used another organism called this is the marine source organism. From that marine organism, we isolated all the three proteins, phycoerythrin, phycocyanin and allophycocyanin in the purified form. And from that again, we again went for the crystallization followed by the X-ray diffraction study, and we derived the structure. And this is one of the structure which is called the phycoerythrin structure. We could get alpha protection, and we noticed that on the alpha there are two what you call chromophore groups are attached at these positions. In case of the beta, there are three such chromophore groups, of which one of the chromosome is doubly attached and other two fruits, like that. And then we could get. In addition to that, in comparison to the phycoerythrin feature of from other organisms, there are slight differences that in the sense that we could also found that a specific insertion of 9 residues of it, and this has a higher affinity for the attachment like that. So like that, even in the phycocyanin also we could get from the same organism, and that was crystallized, and we could purify that. And there also we bought. And it has one uniqueness is that there are 3 amino acids, we call it, this is called phenylalanine glycine, and there is one more also, the 3 amino acids which are supposed to get involved as a evolution. In the sense that if one of the amino acid is absent, other two also will be absent. If one of them is present, other two also will be present. So there is a co-evolution of that was noted. Like that, we also got for the APC structure, but it was a trimeric structure. Then we wanted to make use of these proteins for therapeutic purpose. What we notice that we know that whenever there is a stress to the body, whenever there is a stress to that, it will result into the production of oxidant, or you can say redox oxygens. So ROS are generated whenever there is stress. Our body or our cellular system has a mechanism, whatever some amount of oxidants are produced, they are neutralized by the antioxidant. But they can do that job up to certain level. If the stress is given beyond their capacity, there will be a damage. There will be a damage. So what you require? You require external antioxidant. And there are various antioxidants which are available in the market, which can be used. Similarly, this also we can. So we can see that here what you have. Whenever there is a stress, you can see that lot of what you call unfavorable reactions takes place, and that can create complications. So during the oxidative stress, what we have when there are three possibilities. One of the possibilities is that protein undergo into the degradation. This is what called ubiquitination. So here you have the protein which is getting destroyed. Our protein get also aggregated, so it can cause age. It can have aging, early aging. Our third is that if the damage is not very severe, then it can refold. It can come back to the original form. So there are all possibilities. So what we want to do that means that whenever there is a reactive oxygen species which are generated, these reactive oxygen species can lead into the aging. अब जल्दी बूढ़े हो गए. And this happens also to many persons. If you are under very high stress, you can. So what you require to protect that, you require antioxidant. So antioxidant will work as an anti-aging. What we notice that this particular protein, what we call phycobiliproteins, have antioxidant ability, and therefore this phycobiliprotein can also be used as an anti-aging. Then how do you prove that? There are several protocols available to see the antioxidant. These are all titrometric, spectrometric analysis by which you know that, and with a standard antioxidant you can check it whether they are strong antioxidant or not. To know that it has got a positive impact on the health, we use the organism the way Professor Tiwari uses Drosophila as a model organism. I use C. elegans. C. elegans is also another model organism, and it has got several advantages in the sense that it can be cultivated very easily in the laboratory. It can be stored at -70, and there are various mutants available in the C. elegans. So you can plan your experiment using those type of mutants. So it becomes easier, and it has got a comparatively short half-life. And what it requires only what is known as E. coli as a feed. Their food is only E. coli, nothing else. So you provide the E. coli, so you can handle them in a small petri dish. And it requires a temperature of 20 degrees centigrade for its multiplication. Like Drosophila, like this, so it will also have an adult, but only thing that there are two types of C. elegans. We call it hermaphrodites. That means male and females are available in the same, and there are only males also. So what you require, they will lay the eggs. From the eggs it will come out and it will go through the several generations, what you call L1, L2, L3, L4 and so on, and it will have a lifespan like this, and again it can produce as many as about 300 to 1000 eggs per C. elegans can produce. So you have a large number, and it is easy to cultivate in the laboratory. Therefore, we thought that let's use the C. elegans as a model. Now when I use a C. elegans as a model, the normal lifespan of that C. elegans may be somewhere around 15 days, or you can say that 15-16 days. When you have, so it has a lifespan of about 15 days. When this, so what we do, we have a nematode growth medium, it's in the petri dish, and on which we put the E. coli. There is a specific E. coli OP50, that is used as a what you call food for the C. elegans. So you allow them to have a lawn of the E. coli on the plate and then put the C. elegans. It will show the whole, and this C. elegans will survive maybe somewhere around 15 days or something like that. Now what I do, we add the phycoerythrin along with that. And when the phycoerythrin is also taken by the C. elegans, we notice that the lifespan, instead of having 15 days, it can go up to 17 days, 18 days, even 19 days, depending upon the dose what you give. As you increase the dose, perhaps up to certain level, beyond that it will not. Up to certain level it will give. So with the dose dependent manner, you will find that there is an increase in the, and along with that, there are something called surrogative markers. Using those surrogative markers, you can also again check. For example, one of them surrogative marker is the pharyngeal movements or contraction relaxation. You can make out very well. You can observe them also, either using the dissection microscope or even with the naked eye also. You can use the stopwatch and see how many times it is making the contraction relaxation. At the same time, their locomotions can also be marked, and that is done by their body bending pattern. So looking at their body bending pattern, you can make out how many turns it's taking place. Again, with the help of stopwatch, you can make out. So like that you can, so there are surrogative markers also which can. So how long the C. elegans is active can be noticed in presence and in absence of phycoerythrin. So what you can see that. So this is what we need. So we have the normal lifespan, you have the pharyngeal activity. First day you can see both of them are more or less equal, but when it is on the 5th day, if you look, as the time passes, their mobility goes down. So as the time passes, see in the 5th day you can find that you get a less number of pharyngeal activity, pumping action, whereas in case of P treated it is. So like that. Similarly, locomotion assay. So these are the things which can be, these are called surrogative markers. Now here we are using the C. elegans in a normal condition. You can also provide them what is known as oxidative stress. Externally you can give the stress. One of the type of external oxidative stress is you expose them to the heat. We normally grow them at 20 degrees centigrade. If you incubate them at 35 degrees centigrade, what will happen? It's a heat shock. Or you can expose them to the oxidizing agent like such as hydrogen peroxide or some herbicide. If you expose them to such condition, obviously their normal life will go down. So under that situation, what we found that we have, this is what we did. We incubated the C. elegans without PE feeding at 35 degrees centigrade, and then we also exposed the C. elegans in presence of PE with at 35 degrees centigrade. We noticed that when the PE is provided, somehow they have a better tolerance than without PE. So PE has some positive benefit to this. Similarly, when they were exposed to the, let us say 10 millimolar, so when we give the wash with the hydrogen peroxide, they put them in the hydrogen peroxide for about, with this concentration, either 10 millimolar or 20 millimolar, keep it for about 2 hours, take it out, again wash them also, and have the plate with PE, without PE, and see that you will find that there is always improvement in presence of PE. It means that the phycoerythrin has some positive benefit for the survival of C. elegans. So it gives some benefits. Then obviously, there must be some action. So one of the reasons we thought that it is associated with what is known as insulin-like growth factor. in this particular factor, there is one particular regulator which is involved, that is known as DAP16. This DAP16, though it is synthesized in the cytoplasm, it gets entered into the nucleus and it will bind to such a location, such a way that it will trigger all those genes which are required to cope up the stress condition. That means that DAP16 is essential requirement under the stress condition. But then when it will be able to do, provided it enters into the nucleus and it goes and binds to that such a location. And this happens, but then this is also regulated by the upstream genes. One of them is the DAP2 and the H1. Now what does DAP2 does? DAP2 will go for the production of enzyme, that is a kinase, and that kinase will phosphorylate the DAP16. Once the DAP16 is phosphorylated, that 16 cannot enter into the nucleus. If it cannot enter into the nucleus, obviously the C elegans will not have ability to survive in presence of oxidative states. But if the DAP16 enters into that, then you can have, so you can try to do that. So similarly, another negative regulator is that. So you have a DAP2, then H1 and then DAP16. So this is what happens. So you are able to take. So these two. So what we did, we tried to understand whether DAP16 has anything to do with the P. Okay. First of all, what we did, we got the mutant, and in that case, where DAP16 is coupled with the green fluorescence protein. So you are able to monitor the movement of DAP16 from the cytoplasm to the nucleus, which was possible. And then when we try to do it in absence of P, in presence of P, in both the cases, it does not affect as far as the movement concerned. And that was noticed by using this green fluorescence protein. That was that. Then what we did, we used the one mutant strain where DAP16 is mutated, so it is unable to express. And when the DAP16 is unable to express, it has a normal life in absence. And when you have a P along with that, it shows the increase life. That's what happens. This is what it is expected. It means that DAP16 has nothing to do with the P. They are independent. Had it been the case, if the P is associated with the DAP16, it would not have increased the lifespan in presence of P. Then we also took the mutant of this DAP2 as well as H1. And when these two mutants are taken, obviously when DAP2 and the H mutants are taken, that means that there is no negative control on the DAP16. As there is no negative control on the DAP16, what will happen that it will show much enhanced lifespan. What we have 15-20 days, here the lifespan can go up to 30-32 days. And in presence of P, it is further improved. That's what happened. This is fine. So obviously we notice that DAP16 has nothing to do with this. If it is so, there could be something else. And many of you, all of you are maybe knowing that what is this heat shock factor. It's a transcriptional factor. And this heat shock factors is also responsible to induce the stress response protein, some molecular chaperone. So what we did, we use another C elegans, another state, where in a normal case we have a normal C elegans, and that normal C elegans where we have, you can see that this is what actually the normal life. Then in presence of P, there is an increase in the lifespan. Then we took, what we did, we have a E. coli cell, and this E. coli cell is responsible for RNAi formation. You know what is RNAi? That is RNA interference. Okay. So we could have a E. coli cell which could produce RNAi, double stranded RNAi, and that E. coli cell which is producing double stranded RNAi was given as a feed to the C. elegans. When you have a E. coli containing the interference RNA and that will go into the C. elegans, and it will result into what will happen, that this RNAi will interfere, or it will become that you are able to knock down that particular gene. So SSF factor gene is knocked down. Or similarly, we could also do the knockdown of another gene, what is known as, we will see later on. There is another called the stress protein gene. So you have knocked down that particular, so that this particular gene will not go for the expression. Under that situation, when you have, so what is happening here in this case, that see, this is SSF1 RNAi. That means this SSF1 is knocked down, it is not available, it is not expressed. Under this situation, this is what we have the pattern of lifespan. And under this situation, when you provide the PE also, there is no increase in the lifespan. It means that PE is very much linked with the heat shock protein. So you are able to make out their correlations. That's what happens. And then obviously, always whenever you try to see that, you have to see the level of RNA. And we could see that, we could see the level of RNAi, and in the control, what is the level, and this is in the impression. So this is how we can make out very easily. Now what we did,